Thank you, Mahalaya. And thank you everyone for attending and joining us today for the third webinar in our four-part series on science and risk-based stability practices in pharmaceutical development. Today's topic will be leveraging available knowledge to design appropriate stability studies. Both of our speakers work with the IQ Consortium Science and Risk-Based Stability Working Group and share an interest in using science and predictive techniques to stability to accelerate product development and focus stability knowledge and use throughout the product lifecycle. Jack Pellet is a director in the small molecule analytical chemistry department at Genentech, leading a group of analysts responsible for the progression of projects in early development, as well as in-house drug substance manufacturing support. Jack is a member of the Roche Small Molecule Quality by Design expert team and heavily involved in the risk-based predictive stability community. Prior to joining Genentech in 2010, Jack spent time at Pfizer after completing his PhD in analytical chemistry from the University of Minnesota, Twin Cities. Paul Gerst is a principal scientist in the analytical group at Pfizer Groton Labs in Connecticut. Since joining Pfizer 20 years ago, he has contributed to the development of several drug products. During this time, he has developed keen interest in understanding accelerated predictive stability and the chemical interactions of active pharmaceutical ingredients with excipients. He has also spent time understanding and modeling the moisture permeation through packaging components and using this understanding to predict the impact of moisture and temperature on the long-term stability of the drug product. Outside of his normal day job, Paul does enjoy performing as a freelance trombonist and pianist. So with that, I'll turn it over to Jack to
Yes, thank you very much for the introduction, Brian. We're going to talk a little bit about now today leveraging stability knowledge from risk-based approaches. And again, we are Brian, Paul, and myself are part of the Science and Risk-Based Stability Working Group through IQ, and our mission is to further advance the usage and applications of science and risk-based stability approaches for pharmaceutical products. And we hope by doing that we can expand the understanding and potential applications for these approaches for both small and large molecule products, not only within the pharmaceutical industry, but also with health authorities, and doing so through publications, presentations like these, and presenting strategies and case studies. Hopefully, most of you are able to join us for the earlier webinars where we covered application of these approaches for small molecule and biologic therapies. Again, today we'll talk about leveraging that knowledge to design stability studies, and then hopefully you'll be able to join us next week for our last session on the regulatory landscape of these approaches. So some things that we hope you take away from today's webinar are, first and foremost, the value of stability risk assessments and how they can help you with process and product development, and then how we can translate the knowledge that we get, not only from these stability risk assessments, but from the knowledge gained throughout development into a more lean stability program, and how all this knowledge can help us with package selection and also setting specifications. So the way I like to look at this is when we think about stability understanding is start with the end in mind. And this is very much aligned with QBD principles that are currently being applied routinely for process and product development, as a lot of folks know through like a quality by design approach. So really what we want to know is my product safe and efficacious, but for how long, what kind of packaging, and in what kind of environment. A lot of the information that we can get about what our product needs to be able to deliver over its shelf life can come from the quality target product profile. So that's something that I found very useful to look at. If it's available, to understand what kind of packaging configuration my product's going to need, what kind of minimum shelf life is going to be acceptable, and also where that product is going to be used, what kind of countries that product is going to. Kind of following the theme of a quality by design principle, once we have a QTPP, we need to start thinking about what our critical quality attributes are. And from a stability perspective, we've kind of defined these as a subset of those CQAs, and those are what we call stability-related quality attributes. And so kind of a definition here is These are the physical, chemical, or microbiological attributes of that final product that could change during storage or use by the patient. And then, of course, potentially impact safety, efficacy, or quality. So these are the ones that we want to really focus in on understanding. From there, we need to start thinking about what factors or what inputs could influence those stability-related quality attributes. So what process parameters, what material attributes. So these could come from how it's being manufactured, what equipment it's being used to manufacture. That can impact stability. Excipients or the properties of the drug substance that can have an influence on the stability of the product. And of course, the type of packaging that's used can have an influence on the stability of the finished product. Yeah, there can be a lot of things that could potentially influence stability behavior, and we don't have the resources to fully understand everything. So we need to prioritize those and identify which are the most important ones. And there's a variety of different approaches that can be used to do this. I've listed here some of them. The more common ones, fishbone diagrams, quantitative or semi-quantitative cause and effect matrices, FMEAs, PHAs. So there's a lot of examples actually in ICH Q9 that can certainly be applied and have been applied by different companies. I'm going to give an example of a semi-quantitative cause and effect matrix because that's worked well in our organization, and I thought it would be helpful to run through one with everyone. And then, from this, the risk assessment and then the studies that are done to understand the relationships between the inputs and the stability-related quality attributes, you can start to even think about creating a design space that may look a little bit, non-traditional. For example, we usually might say something like the product is stable for at least 36 months at controlled room temperature when stored in 30 count 60 cc HDPE bottles. But from a design space perspective, our product might be stable, acceptably stable in other configurations too. So you could think about, writing it, something like stable over at 0 to 30 degrees C for 36 months with the following attributes. A bottle with a certain contact resin with a moisture vapor transmission rate that's less than a certain amount of water per meter square per day per gram of tablet, as long as the tablets have a certain ingoing water content at release. So you can potentially accommodate a variety of other packaging configurations and in a more of a design space way. And then, similar to process and product control strategies, we can think of having a stability control strategy. So what controls do we need to have around the packaging? How do we set specifications so that we'll have a stable product? Environmental controls, that could be during packaging. Of course, if excipients, attributes of excipients are important, we can develop specifications around those. We might have specifications, or we might have restrictions on the label too, protect from light, for example. And then finally, a verification program that the stability of the product is acceptable. So an ICH type stability study. So these are all things that could be thought of as part of our stability control strategy. So I'm going to go into a little bit more detail now about a stability risk assessment. And the main goal here is to utilize collective knowledge about stability behaviors and liabilities so that we can identify the risks, and then again, prioritize our experimental studies and ultimately develop a control strategy. So this word collective is important. You know, we don't want this to be done by just somebody from Analytical or somebody just from formulations. There's a lot of folks who are going to have knowledge not only about this product, but similar products, and we want all their input into this risk assessment. Let's get a lot of questions too. When's the right time to start a stability risk assessment? You know, for a small molecule product, I find it best after that formulation's been selected. Although you can think of doing, you know, lean kind of risk assessments to help guide that formulation selection. The one that I'm going to describe here is probably most suited once you have a formulation in mind. Another important thing is you want to do this proactively. Oftentimes, you know, there'll be an issue, and then people say, oh, well, maybe we should do a stability risk assessment to identify the root cause of this issue, and that's fine, but it's a lot more. It's a lot better, as you can imagine, to be able to do this proactively to eliminate those issues or reduce the frequency at least. This is not a one-time exercise. It's important to revisit, and if you're doing a scored risk assessment, look back to see if the score should change. You will be accumulating additional knowledge throughout development, and so your risks and areas that you may want to further evaluate could change. So in these approaches, we can do these risk assessments not only during clinical development, but also commercial development and post-approval. So the first step in a risk assessment like this is to figure out what your potential stability-related quality attributes are. And for a small molecule product, this is, you know, these are some examples. Assay hardness, dissolution, you know, degradation is a common one. It could have to do with microbial growth, appearance, you know, form change, maybe something to think about for the product, and water content or water activity. The next step is, in a semi-quantitative approach, is to weight these attributes in terms of importance, and what we think of them as important in terms of how likely they are to be shelf life limiting. This example here is a 4-tiered approach, but you know, other companies have used 3 tiers approaches with just 10, 5, and 1 systems. Those work just fine too. The ultimate goal here is to be able to, you know, have some separation, though, in the scoring, so be able to weight them, you know, so that all attributes aren't weighted the same. So in this case, we might give a 10 for something that's very likely to be shelf life limiting, a 7 if it might be shelf life limiting, but maybe we haven't really studied it that much. We're not really sure. It's a 3 if it's, you know, likely to be something that's changing, but we unlikely to impact the shelf life, and we really haven't studied it or don't have a lot of knowledge around it. And then if and then the shelf life is not shelf life limiting. Okay. The next, so this is an example of all a set of different stability-related quality attributes and how they might be scored. It's important too to provide some comment or rationale for the scoring, so that when a project's handed off, that knowledge isn't lost. And so that's just one really important part of a stability risk assessment, is to try to capture that rationale concisely for how it's scored. And so the next step is to list out, you know, all the inputs, those process parameters, material attributes, excipient attributes that could be packaging, that could impact or potentially impact one or more of your stability-related quality attributes. And so, you know, here's some examples for drug substance attributes. The salt stoichiometry can have an impact on stability, how much amorphous the particle size often does. So you can read these, but you want to have a pretty comprehensive list to start with of everything that potentially could impact stability for the drug substance, excipients, packaging, and manufacturing process. So just another example of how this could be constructed using more of a fishbone diagram approach. This is something that a lot of folks are familiar with when doing risk assessments, and this just gives an example of other attributes for small molecule products. It might be a more of a amorphous spray dry dispersion kind of product, or a crystalline API product, or a solution product, where you might have different things, different inputs that are going to be potentially more important than others depending on the product. So it is going to be very product and formulation dependent what attributes you want to assess. And then the next step is to look at how likely those inputs, those material attributes, process parameters are going to be to influence those stability-related quality attributes. So we'll score those. In this case, it's a three scored system, whether it's known to have an impact, so known to have a relationship between that input and that stability attribute, and possibly it's been observed in previous stability studies or products of very similar nature. A 5 of it may have an impact, but haven't really studied that extensively. Or 1, it's there's really no, it's very unlikely there's not going to be a relationship between those. So I'll spend a little time here. This is an example of a constructed cause and effect matrix and how it all comes together. So up top here we have the scores that we assign for each stability-related quality attribute: 7, 3, 10, or 1, for example here. This next row just shows their relative importance. And then down the rows here are all of our different input sources, and we've gone ahead and scored the relationship between that input and that attribute. So this is for each stability-related attribute. Appearance, for example, and the salt stoichiometry has no impact on the final tablet appearance, so that was a 1. And for assay, it was also scored a 1. You can see here that we've got a 5 for degradation. And then what happens is each of these is multiplied together, so that's 7 times 1 plus 3 times 1 plus 10 times 5, etc., and you get a final total score. And in this case, it's a 109. So you do these for each input source, and you get a whole list of scores at the end. And the absolute score doesn't really matter. It's, you can imagine you'll have different number of attributes. So if you have more attributes, the score is going to be higher. That doesn't mean it's a higher risk product or anything like that. So this is just to help prioritize which are the highest risk attributes for your, or inputs, relationships for your particular product. So then, of course, these can be sorted in terms of highest score to lowest score. So we've done that here. In this case, the polymorph of the drug substance had the highest score across these different attributes. And another thing that's really nice about this approach is that it's all in one place. It's visual. You can see, why it's scored high, which attributes are most likely to be impacted by polymorph, which are the most important ones. So it can really help you identify which areas you need to study. And then you can go through it and say, what, these are going to classify these 8 inputs as the highest priorities, the ones I really want to study. I see a lot of 10s in here. There's a break point after 174 that I'm going to call these my high priority relationships to understand. These are my medium priority, and these are my lower priority. And then the next step here is to agree on how you want to go ahead and study. What are you going to do with these medium and high functional relationships? So some of them you may want to start studying experimentally right away. And by doing this early, you can work with your formulator to try to incorporate some of these variants into stability studies. And then you can also use that risk assessment to say, you know what, I don't need to test every single stability-related quality attribute. I'm going to focus on degradation for this relationship. Some of them you may say, you know what, we know these are potentially high risk, but right now we've got good controls on the excipients. Additional variants may impact stability, but we don't have those right now. So we're going to go ahead and park those studies or that manufacturing equipment. And when we do have that available, we'll study it. And similarly, some things you may just say, we're going to set controls on it right now. So we'll restrict our process ranges or the material use. It's easy enough to do that. And then a fourth thing you can do is modeling. And there was some talks earlier in the last couple of weeks about what modeling approaches can be used to understand these relationships. So with that, I'm going to go ahead and turn it over to Paul, so he can talk to us about the packaging and lean stability.
Thanks, Jack. So that modeling is a really good segue into the next portion of this talk. So if you were able to attend some of the two previous webinars, you saw that that predictive stability is a big part or a major tool in stability and understanding. And when we combine that with the risk assessment, we can really come up with well-sounded predictions and models to explain and understand what's going to happen and predict everything ahead of time, so that when it comes time to perform the actual stability studies, we hopefully won't have any surprises. One of the most important things that we can model is the effects of temperature and humidity. Now, when products are set up at a particular storage condition, 25 or 30 degrees, that temperature condition remains constant throughout the entire study, with the minor fluctuations that are allowed. But the internal relative humidity of our packaged material does change over time. One thing about the packaging is that when we go through the risk assessments and we determine which attributes are the most important, what are the ones that we can actually control. Most of the time, packaging is a big component of that risk assessment and any mitigations that can come from that. For example, let's say that you were going through that risk assessment and you would determine that light was a major implication to stability. Well, that clearly tells you that you will not be putting the material in a clear blister where it could be exposed to light and degrade. You would probably put it into something that was far more light protective. Again, packaging is a big component of that. If you look at all the food products that we have, in any of the pharmaceuticals, there's a lot of science that goes into that packaging. It's not just picked necessarily just for show. So coming back to humidity here, besides light, temperature and humidity are some of the biggest factors that can really impact stability. And where we can control the temperature, the humidity is variable over time. And we can model this very well through permeability studies. I'm not going to go into a lot of the technical details because there's just not enough time in this short webinar, and there's a lot more to discuss. But we know that the moisture vapor transmission rate is variable depending on the difference between the internal relative humidity and the external relative humidity. So whereas we set a constant temperature condition, the external humidity is kept constant, but the internal can change depending on how rapidly the moisture transmits through the bottle. Next slide. Yeah, next slide. So, no, go back. Yeah, there we go. So now that we know, once we know the moisture permeability properties of a package, and we know how much material that we put into the package, we can apply a lot of the first principles of moisture equilibration to predicting what the dynamic water activity or relative humidity would be inside a bottle. For example, the more mass that we have inside a bottle, the more that water will be distributed within the weight gain of the tablets rather than the head space. We also know, based on different material properties, how much water they will absorb versus a different formulation. So we can put this all together to fully understand the ultimate equilibrium inside that container and model and know exactly throughout the entire course of stability or the stability time frame what that internal relative humidity will be. And we can, by knowing how humidity and temperature will affect the stability, we'll know exactly what's going to happen. And let me show you a couple of examples of this. So here are a few different types of packaging. And depending on the bottle size or the shape, whether or not there's a desiccant, whether or not you have a foil blister or a PVC or an Aclar blister, we know what the moisture permeability properties are. We know how much mass we're putting into the bottle, and we can predict out exactly how much humidity there will be inside the bottle. So depending on whether or not you package in the 60 cc HDPE with a 30 count or 15, or you add desiccant, you can see that over the course of 3 years, the humidity range can be anywhere between just under 20% all the way up to 75 or 60%, depending on the storage condition that you put it in. So humidity has a wide gamut, and understanding this is in scope of the risk assessment is extremely important. Next slide. And here's an example of how we can put all this together. The left plot shows the internal relative humidity as predicted by our models. And the right side shows a graph of a particular shelf life limiting attribute, in this case, the percent of a degradant, that will increase with response to the changing humidity. So during the risk assessment, we perform these risk assessments without considering what that packaging is going to be. Once we know that the humidity is going to impact, or any one of those attributes is going to impact the stability, we go on to the mitigation strategy. So this example, which is one of the more common ones, is the effective humidity. And this can be applied to both chemical and physical stability. In this case, I'm showing a chemical stability problem. So when we perform the risk assessment, we consider an open condition. They're perfectly exposed to the atmosphere. That would be that purple line. You see that in a 75% relative humidity condition, the degradation grows very rapidly and would surpass the specification in only about a half a year. Well, because of the risk assessment process and all the work that went into understanding, we know that we'll have to mitigate through packaging. In this case, the blue line is showing what happens when we put the material into a 60 cc HDPE bottle. It limits the humidity a bit, and the percent degradation has been reduced a little bit, and we can get a little more than 1.5 years. But that's not good enough, and so we can iterate this further by adding 1 gram or 2 grams of desiccant, and we can understand exactly what that packaging effect will be. Now, this example, as I showed you, it just shows a percent degradant, but physical stability is often an important factor as well, that sometimes can be modeled either by first principle models or empirically. Most of the time, you might have a certain threshold of a humidity value that you want to stay away from because you know there's a form change above a certain temperature and humidity condition. So using this packaging knowledge, again, Jack had mentioned an amorphous material earlier, or polymorph properties earlier. Well, if you know that a certain API is subject to a certain change at a certain relative humidity, you can use this packaging mitigation and the modeling to help understand and limit the humidity that you would see over the course of time. Next slide. All right, so all this goes into now what we refer to as the lean stability strategy. And this isn't necessarily something that's performed at a at a certain gold star period of time. This is an approach that occurs over the product life cycle that basically leverages all the information that you have, either from clinical stability, force degradation, any experiments that may occur to understand humidity, and brings us all together to understand what are the most important aspects of stability to measure and control. And this also helps to limit the amount of testing that we do, so that we're focusing on the most important aspects of stability, so that we can expedite our products faster without, and this is the most important part, impacting the safety, efficacy, or quality of the product. And that's in red for that very specific reason. We're not trying to perform less stability, and often we perform more stability. We just want to make sure that we are performing the most important stability tests that to make sure that we maintain that safety and efficacy of that product. So where can this idea of lean stability strategy be applied? Well, really, it's throughout the entire life cycle of the product, from very early on, and Jack mentioned that the stability risk assessment is performed just after formulation. And this can go all the way through post-approval. And of course, as the project progresses, we learn more and more and more, and we can apply better and more confident knowledge to coming up with an ultimate stability strategy down the road. Now, it is most appropriate for stable and well understood products, and I'm going to describe what stable means in a moment. But really what we're looking for is that we want to demonstrate this on products where we know that we have a good understanding of the degradation that's going to happen. If you have a certain aspect where there's a lot of variability and you can't understand what's going on, then that particular attribute may not, or product may not be a candidate for lean stability, because you just don't have the full hold on the stability knowledge that you need. And as I said, it was written in blue, I pretty much mentioned already. It may mean more work now, but it's meaningful work that you're leading up to throughout the product. So you're basically focusing on what could be important now, what might be important, what you've seen to be important, and then you're going to define later on, as you go throughout the stability life cycle, that you're going to focus on the most important attribute possible. Early on, you may be testing you may have a certain test, for example, water content, but you will learn over time that water content doesn't provide any meaningful data, but there's something else that's probably more apt to limiting stability. So this process, this approach takes you through this. Next slide. So I want to define what a stable product is. A stable product does not mean that there isn't a change. In the previous example I just showed you, an open dish condition, the degradation increased very, very rapidly. That doesn't mean it's not a stable product. A stable product means that once we applied all the mitigation strategies and that we understand exactly how that product is going to react with different environmental variables, and we can predict and control, a stable product is one that has a very low likelihood of actually surpassing a specification that was set. So as I said, I think as I was talking, I think I said the same. What's in that second bullet is that a product can be stable if whatever change that is observed is limited, controlled, and predictable. And if you look at everything that's been talked about throughout these webinars, that's really what we're doing. We're looking to increase our ability to predict the changes that are going to occur, so that we can define a certain product in a certain configuration as being stable. Next slide. So what are some of the advantages of lean stability? Well, first of all, there are big advantages on the industry. And the fourth bullet here says fewer product surprises and failures, but honestly, I think that's really the very first one. The more knowledge that we can learn ahead of time, early on, by performing more experiments, even if there's a lot of work, again, this is not designed to save work now, it's more work now, it leads to fewer surprises and failures down the line. So if you're in early phase 1 and you're performing this and you're starting to build your knowledge, hopefully by the time you get to phase 3, that you are not seeing something that you've never seen before in your stability. It also leads to more efficient product development, because the testing that goes on is testing the most important aspects of the product, so that you gain the confidence early and quickly, so that you can actually get the product out into the clinics earlier without having to wait 3 or 6 months for the data. You can get that knowledge much earlier with higher confidence that you're not going to surpass the specification. And then that goes into maintaining the safety and efficacy of the product. It's good for the agency as well, because we're providing meaningful data to the agency that tells them we understand what our product is. We're not just testing everything because we don't know what's going on. This is what we're testing. This is why we're testing, and this is what we're learning from it. So it helps to build confidence with the agencies that we understand what our products are doing, and that we have a good handle on them. It also leads to, again, this is a benefit for us as well, fewer product recalls due to stability concerns, which then leads to a lot of explanations and other discussions with agencies. And finally, for patients, there's a big benefit for patients. First of all, because we don't have to wait a long period of time to fully understand the stability, because we have a more methodical approach, we can get the products to the clinics earlier, get the trial started, and it really helps shorten the timelines for development. Not only that, but it provides higher quality products, which means when a patient opens up that bottle, they're not the product that they had a year ago is going to be the same that they have now. It's not going to look any different. It's not going to have any issues that they're going to question, and so that they don't end up questioning, why is this tablet look like it's falling apart, or whatever it happens to be. And most importantly, and this goes back to what I said before about recalls, is that it keeps that supply of medicines going. We've heard, over the past 10 years about products being recalled, and that that product is no longer available to a patient, which then now they have to find something else that works or may not work as well, and they really they really found the product that they were on was working for them. So this really does have a lot of advantages, that the more that we understand our knowledge now, the better off people are going to be who are receiving this product later on. Next slide. Okay, so here, so here are some examples of lean stability strategies. So really, what we're doing is we're leveraging a lot of these tools, and you're seeing in the predictive stability work that you've seen in the previous webinars, and the risk assessments are all part of the tools that we use to allow us to start these clinical trials earlier. And the stability risk assessment, accelerated predictive stability, even applying certain statistical approaches to understand the data, or even setting specifications, such as what's outlined in Q&E, helps to provide that confidence in that information earlier on. And I think a lot of this is repetition of what I already said, but we really use it to enhance that knowledge of our product, so that when it comes to time for marketing applications, that we're applying the most important stability pull points and tests possible, so that we're not overloading our labs with needless testing, and that we can take a more lean strategy to that stability without impacting, again, safety and efficacy. And a lot of these, and I'll show you an example in a moment, is already outlined in ICH Q&D. But again, in order to do this, you need to have that knowledge ahead of time, so that you have the confidence that when you apply the strategy, it's going to work and maintain those qualities aspects that I mentioned before. Okay, let's go on to the next slide. So here's an example of a bracketing stability design. Again, this is outlined and taken directly from ICH Q&D, where you might have 3 different strengths, 50, 75, and 100, but you can bracket that 75 by looking at the highest and lowest strengths. Again, there's a lot of knowledge that goes behind that justification. For example, similar formulations, or for example, that 50 milligram may be maybe a slightly different formulation that has a higher probability for degradation potential than the 100. So now you're bracketing that through. Another example is shown in the lower right, this 1/2 reduction, where you might have different batches, and at different time points, or 2 different strengths, 3 different batches in each strength, but you're not testing throughout the entire. You're not testing every strength in every batch. You're skipping, and you're performing what's called skip testing, or just reducing, and focusing on certain batches and certain strengths at one in time, and building that matrixing data throughout. So this saves a lot of testing. Not only does it save a lot of testing, but as you test more and more and more, and especially if you're testing things that you really don't need to, you always open up the opportunity for an investigation, not because there's a problem with the product, just because of things that always happen in the labs. Murphy's Law, this is going to go wrong, it's going to go wrong. So if you can avoid excess testing and mistakes, because that's often what happens when you're testing a lot, applying these type of bracketing strategies may go a long way, again, and I'll say it again and again, without impacting the safety or efficacy of the product, because we have already performed the proper experiments necessary to understand the product. All right, next slide. So this is an example. So this is basically a what if example, where we looked at 28 different products that had full stability, a full matrix at all the testing, and looked at what would happen if we made our assessment based on a 2/3 or 1/3 matrix, basically taking 2/3 of the design points out, or 1/3, and then using the remaining data to predict the shelf life. How does that compare to a full set of data? And in short, what you're seeing here is that the data is pretty similar to a full matrix, but if anything, it's providing a little bit more of a conservative estimate. So it's above that line, not below it. And there's very little difference. As you can see, there's a lot of overlap between a 1/3 and a 2/3 matrixed approach. So this just goes to show that this type of strategy will certainly not impact the final conclusion. Next slide. All right, so in this slide, so the use of statistics is a big one, and this is noted in ICH Q&E. And basically, it's applying all the knowledge of predictive stability, clinical stability, registration stability, using the approaches with statistics to understand what the data is, to understand what the trends are, justify bracketing and matrixing, and even justification of certain specifications, or justification of removing a certain test just because of the solid data that may exist. And let me show you a couple examples of where this could be used. Next slide. So here's an example for testing the assay for drug substance. And looking at this data, the slope is not significantly different from 0, and this is applying the Q&E, applying the statistics, and there's a good confidence that 99% of the tests are going to be within 98.6 and 102, so that we know that from this data that the process and the stability is well controlled. So what this tells us is that this data, by applying statistics, is a good candidate for elimination of assay on stability for commercial. Next slide. So here's another example for drug product. So now if we apply this the same type of strategy for assay, and we apply the statistics to multiple lots, and there's quite a few different lots and conditions here, that essentially what we can do is that we've demonstrated that there is no change on assay over time. So this would fall into one of those non-value added tests. So then the question would come, well, where's the control for assay if we're not testing on stability? Well, really what that is that we would test it at release, and you can see it's underlined at the top there is at release. So if we test assay at release, then that helps to guarantee that throughout the life cycle or the lifetime of the product, that the assay will not change. Again, we have the data that supports it, and we know if we keep the assay, the release value within a certain value, even if there's a little bit of change, the probability of that surpassing a specification is very limited. So again, this is what we could use this data to propose that we don't measure assay either as often or not at all. Next slide. There are some challenges, and I see we have about 10 minutes left, so I think there's only a few more slides here. There are some challenges in implementing lean stability. Again, this is an approach. It's not just a single thing that we do, but it's a mindset over time. So there is an investment early on to do additional work early on to enhance that knowledge. And there may be some, because a company may be limited by resources, either by equipment or product or people. It may be difficult to achieve that level of stability knowledge. It says no clear framework here or process for conducting stability risk assessments, and that's true and somewhat not true at the same time. If Jack has outlined what we typically do for stability risk assessments. So one of the challenges is applying this in your company as a way of conducting these risk assessments. And of course, there's always the fear of questions or rejections and non-acceptance by health authorities. Approaching this lean stability idea often comes back with, well, we might get a query on that. That's often something that you hear. We might get a query. And the response to that is, okay, let the queries come. The whole point of this is that you're gaining that knowledge early on so that if a query does come, you have the confidence that you can fully answer that and justify further why you're taking a certain approach. And of course, the biggest challenges are by the regulatory reviewers. We take the scientific based approach, but it needs to be accepted by the regulators. Whereas there's some acceptance by certain health agencies, it's not always accepted by all. And the only problem is that although ICH Q&A and Q5C are there, they don't necessarily promote the use of more modern risk-based approaches that we're talking about here. So one of the goals in this webinar series is to increase the knowledge of what we're doing and why we're doing it, and provide better and more thorough scientific justification to hopefully discuss and talk to these regulatory agencies of why the approaches that we have are appropriate. Next slide. So finally, we conducted a survey of 19 pharmaceutical companies to just understand what their approaches are for stability risk assessments. So actually, I'm going to take a pause here. I think, Jack, I think you wanted to talk a little bit about this, right?
Yeah. So this is just example, I think, from a survey that was conducted a few years ago about uses of lean stability approaches and risk assessments. And so of the 19 pharmaceutical companies that are surveyed, 16 of those indicated that they are conducting stability risk assessments in one form or another. How they're conducted, what kind of tools are used did vary, quite broadly. Only about 25% used a formalized scoring system for doing their assessments. Majority of them used more of a general assessment for doing these kind of stability risk assessments, and that's okay. I mean, it really is a bit phase dependent in the purpose of it. And what was also interesting is that over 50% of those who were surveyed actually communicate the results in the regulatory submission. So this can be a really powerful way to communicating your confidence in your stability understanding by summarizing the outputs of those risk assessments in the regulatory submissions. So I'd encourage you to take a look, a little bit more detail in this paper here. It came out of the IQ group on how these lean stability approaches are being used and incorporated into regulatory submissions. Yeah, so just to wrap things up, we hope that you've been able to get a better appreciation for the utilization of stability risk assessments and how it can be used to identify and prioritize risk with products, and then how that knowledge can be and should be used to support more lean, efficient stability strategies all throughout development and commercialization. And with that, thank you for your time. And Brian, I think we're ready to entertain questions.
All right. Thank you guys very much for an excellent webinar. So we did have some questions that have come in. One of the questions from early on is around the stability risk assessment. What is the criteria to define if some RPN values fall in high or medium risk category? So Jack, I think that's directed to you.
So if there is no defined score that would, classify it as a medium or a high risk. At least the approach that I provided as an example, what we would look at is it's a bit subjective, but we'd look at if there's a break point in the scoring that would say, these are clearly the more important ones. We might also look at the ones that have tens and try to include those in the ones that we would classify as worthy of highlighting as high priority. So there isn't a defined, at least with this approach, a defined threshold.
Excellent. We had a slide that came in or a question on slide 26. And this was centered on, do you think that predictive stability could be applied to percent of crystallization in an ASD tablet?
Yep, I saw that question come in as I was talking about this slide, and I hope I covered it, but during when I was talking. But basically, yes. In these cases, a perfect example is that we may know that above a certain relative humidity, that there could be crystallization or a form change, hydrate change, whatever that particular change in the API is. And then we can use this knowledge to ensure that we stay above or below a certain relative humidity, depending on what it is.
Excellent. And then a question around, would you use a tighter range at release to ensure that the product would be in specification over its shelf life?
Yep, I think I touched upon that when I was, that's on a later slide. Let's keep going, a couple more. That one right here on the drug product. So yeah, essentially that's what the goal here would be, that once you have that data and you knew what the particular trends and the spread of the data was, that you may have to tighten that assay instead of it, because you certainly wouldn't want to allow a release assay of 92% if the 95% confidence interval it looks like it's going to be a little bit below that after 3 years. So yeah, you could certainly tighten the specification to ensure that the error bars stay within, and that's another part of the statistical process here is using this data to help justify a certain spec, or even tightening it if necessary, in order to meet the long-term goals.
Excellent. Let's see, one last question before we get to the top of the hour. How does one handle a major photostability risk and low thermal and thermal humidity risk in terms of its lean stability approach? Would this still be a suitable candidate for lean stability or not?
I think that would depend on what you knew about the control of that. So coming back to an example I gave before, if you know that your if your product is extremely photosensitive, one of the mitigation strategies to that may be putting it into a foil blister. And if you do that, then that risk of light has now been mitigated. so that you are that's no longer a consideration for something that would impact. So again, with that being a lean stability is that process where we're applying the stability knowledge, the risk assessment, and the mitigations together to ensure that we have a predictable, so that the degradation that we have or whatever the change does become predictable. And the same thing would be for the thermal and humidity risk as well. In certain cases, so one of the things that, and I didn't touch upon this before, that as we go through this process and we understand more, it may lead to the fact that we need a different a higher specification for a certain product in order to meet that long-term objective. If a default specification of 0.5% or 0.2 for 0.5 for a qualified impurity or 0.2 for an unqualified impurity is there, and you're finding that throughout all the mitigations strategy that you did, that you won't be able to make a certain you won't be able to meet that specification, then you may have to that may be the trigger to further experimentation, in other words, safety studies, to increase that particular limit so that you can now get that within the limit of your long-term goals. So yeah, it would still be a suitable candidate for lean stability, because again, lean stability is the application of this process to reach the goal of knowing how to control and predict that in the future.
Okay, excellent. And I think there are some other questions that have come in. We will be taking these questions and creating a frequently asked questions document that we'll share with the attendees. But thank you very much for attending today, and thank you both for an excellent discussion and great presentation. And we'll hopefully see a bunch of folks back next week for the last webinar in our series. But thank you both very much.